Hello dear students. Today we will continue with chapter 1, transformation of food into nutrients, digestion, but we will solve some typical exercises. You should have with you a notebook and a pen, your support guide and a phone or a laptop. Please sit in a quiet place, prepare your tools, stay optimistic during study and take notes. Watch the video more than once and ask your teacher for more clarification. You have to study the course well before you start solving the exercise. Read the questions carefully. Underline all the verbs in the questions to indicate how many parts the answer must be formed of, and recognize how to answer each type of verbs, whether it needs or doesn't need justification. We will start with exercise 14, page 14 of your biology support guide. Amylase is an enzyme that digests starch. This enzyme is produced by the salivary glands, salivary amylase, and by the pancreas, pancreatic amylase. A grade 9 student has two bottles, A and B. One of them contains pancreatic amylase while the other contains salivary amylase. In order to identify the type of amylase contained in each bottle, he performs the experiments shown below. There are two experiments, experiment 1 and experiment 2. In experiment 1, there are two tubes, tube 1 and tube 2. Tube 1 contains 10 grams of starch plus amylase of bottle A and NaOH solution. Tube 2 contains 10 grams of starch plus amylase of bottle A and distilled water. However, tube 3 contains 10 grams of starch with amylase of bottle B and NaOH solution, and tube 4 contains 10 grams of starch plus amylase of bottle B and distilled water. Referring to your acquired knowledge, describe the test that permits to identify the presence of starch in food. When you are asked to describe an identification test, you have to learn it very well from your guide and from the previous videos. You have to write the steps and write the expected result of the test. So to identify the presence of starch, which test do we use? We use iodine test. So we add few drops of iodine solution to the food. If the color changes from brown to dark blue, so the food contains starch. So this is the expected result, if the color changes from brown to dark blue. Give the name of the digestive juices containing the enzymes mentioned above. The digestive juices are the juices in our body which contain the enzymes. So salivary amylase is present in the saliva. So the saliva is the digestive juice which contains salivary amylase, and the pancreatic juice secreted by the pancreas contains the pancreatic amylase. Indicate the product of the digestion of starch after the action of amylase. Of course, you have to know that after starch is digested, it is divided into smaller molecules which are maltose molecules, and the verb indicate doesn't require any justification. After 2 hours, the student obtains the results shown in the table below. So as you can notice in the table, after 2 hours, tubes 1 and 4 still contain 10 grams of starch. However, tubes 2 and 3 contain no more starch. Referring to the two experiments and to the table above, analyze the two experiments. The verb analyze. When the verb is analyzed, you have to mention all the conditions of the experiment, including the temperature, the solution present in the tube, the kind of substrate used, the kind of enzyme used, all the conditions present in the experiment, and you have to relate them to the results which are presented in this table. In experiment 1, the two tubes 1 and 2 contain 10 grams of starch and amylase of bottle A. but NaOH is added to tube 1 while water is added to tube 2. The quantity of the starch, this is the result. The quantity of the starch remains constant at 10 grams in tube 1 while it decreases to 0 grams after 2 hours in tube 2. While in experiment 2, the two tubes 3 and 4 contain 10 grams of starch and amylase of bottle B. However, NaOH is added to tube 3 while water is added to tube 4. The quantity of starch decreases to 0 grams in tube 3 while it remains constant at 10 grams in tube 4 after 2 hours. So it decreases to become 0 in both tubes. The quantity of starch decreases to become 0 in both tubes 2 and 3. However, it remains constant at 10 grams in tubes 1 and 4. What can you deduce about the type of amylase contained in each bottle? When the verb is deduce, you have to derive a conclusion by reasoning. You have to mention the conclusion that you reached and you have to give the reason that led you to this conclusion. So bottle A contains salivary amylase since it acts in a neutral medium, while bottle B contains pancreatic amylase since it acts in a basic medium. Remember what is the medium of the solution which is saliva present in the mouth? It is the neutral medium. So this medium contains saliva, which means that it contains the enzyme salivary amylase. However, the solution which is produced by the pancreas, which reaches the intestine, is the pancreatic juice that has a basic medium. So this enzyme is the pancreatic amylase, which is present in the basic solution of pancreatic juice. We will start now with exercise 30, page 27 in Biology Support Guide. This is an official exam, first session of 2017. In the framework of studying one of the functions of the small intestine, the quantity of nutrients is measured in this organ as well as in the blood leaving the small intestine after a meal. The results of the performed measurements are represented in the adjacent document. You have to study this document well before continuing the exercise. As you notice, there is the time, which is the manipulated variable, and there is the quantity of nutrients, which is the responding variable. How many responding variables do we have? We have two responding variables, the quantity of nutrients in the blood leaving the small intestine and the quantity of nutrients in the small intestine. Draw a table showing the variation in the quantity of nutrients in the small intestine and in the blood as a function of time. So as a function of time, this means that time is the manipulated variable. Time is the manipulated variable. You have to put all the times presented, time 1, time 2, time 3, and time 4. And the responding variable is the quantity of nutrients. There are two responding variables. The quantity of nutrients in the small intestine, you have to mention the values at each point of the time. So it was 90 at T1, 60 at T2, 20 at T3, and it gets to be 2 at T4. However, in the blood leaving the small intestine, at T1 the quantity is 10, at T2 the quantity is 40, at T3 it increases to 80, and at T4 it increases nearly to 98. You have, of course, to remember that you have to write a title for each table you draw. So the table showing the variation in the quantity of nutrients as a function of time after a meal. Of course, you can get the title always from the question. Analyze the results shown in the adjacent document. Let's talk a little bit about how to analyze such a graph. In this graph, there are two responding variables. The quantity of nutrients in the small intestine and the quantity of nutrients in the blood leaving the small intestine. I start my analysis by comparing the beginning point of each curve. So the quantity of nutrients in the small intestine at time t1 was 90 greater than 10 or 10 less than 90. I have to compare the beginning point of both, and then I continue with the variation of each one. How is it varying? Is it increasing? Is it decreasing with respect to time? So at time t1, the quantity of nutrients is 90 arbitrary units at the level of the small intestine, which is greater than, at the level of the blood leaving the small intestine, 10 arbitrary units. This quantity decreases from 90 to 2 as time increases to reach T4. On the other hand, or on the contrary, it increases from 10 arbitrary units to 98 arbitrary units at the level of the blood leaving the small intestine during the same duration, which is from T1 to T4. What do you conclude concerning the intestinal function revealed in the document? Now, after we have analyzed the graph and we noticed that the quantity of nutrients is decreasing in the small intestine and increasing in the blood leaving the small intestine, we have to conclude the intestinal function shown by that graph. So the revealed intestinal function is the intestinal absorption. How did we know that? Since the nutrients are moving from the small intestine to the blood leaving the small intestine, we know that absorption is taking place. Indicate another function ensured by the small intestine. Any other function is enough. So the other functions might be chemical digestion by digestive enzymes, or mechanical digestion or passage of food due to the peristaltic movements. You should have memorized these parts, in addition to the characteristics of the surface of exchange of the small intestine. These are acquired knowledge. So list the characteristics of the surface of exchange of the small intestine. The characteristics are that it has a thin wall, it has a large surface of contact or of exchange, and it is highly vascularized, meaning that it has large number of blood vessels. This is it for today. Have a nice day.